Welcome to this lecture on parasitology. Today, we are going to explore the fascinating and sometimes gruesome world of organisms that survive by exploiting others. We will look at parasites, which steal resources, parasitoids, which consume their hosts entirely, and vectors, the living bridges that transport diseases. Understanding these creatures is not just about biology, it is about saving human lives, protecting our agriculture, and understanding the complex evolutionary arms race that shapes our natural world. Let us dive in. To begin our journey, let us understand what a parasite actually is. Imagine an uninvited house guest who eats your food but never pays rent, being careful not to destroy the house so they aren't thrown out on the street. In biology, a parasite operates exactly like this. It lives on or inside a host, stealing nutrients and shelter. While it causes harm, it usually avoids killing the host immediately because it needs the host to survive and reproduce. This one-sided relationship is crucial to study because parasites cause some of the most devastating diseases in human history, like malaria. They also play a massive role in ecology, constantly driving an evolutionary arms race between their evasion tactics and the host's immune system. Now that we know what parasites are, how do we measure their spread? Epidemiologists use a mathematical concept called the basic reproduction number, denoted as R0. Conceptually, this tells us the average number of new infections caused by a single infected host in a completely susceptible population. The formula is R0 equals beta times C times D. Beta is the transmission probability, meaning the chance the parasite jumps during an interaction. The variable C is the contact rate, or how often hosts interact. Finally, D is the duration of infectiousness, which is how long the host sheds the parasite. If any of these variables increase, the parasite spreads faster. If R0 is greater than 1, we get an outbreak. If it is less than 1, the parasite eventually dies out. Let us see this formula in action with a concrete example. Imagine a village where a child is infected with an intestinal worm. We are given that the transmission probability, beta, is 0.1. The child has a contact rate, C, of 5 close interactions per day and remains infectious for a duration, D, of 4 days. We need to find out how many new infections this child will cause. We choose the basic reproduction number formula because it perfectly models this scenario. We substitute our values, multiplying 0.1 by 5 and then by 4. This step-by-step multiplication accounts for every factor driving the spread. The final answer is 2. Physically, this means the infected child will pass the worm to 2 other children. Since 2 is greater than 1, the parasite will continue to spread, indicating an urgent need for intervention. Following up on that alarming result, let us see what happens when a local clinic intervenes with a fast-acting deworming medication. The transmission probability and contact rate remain exactly the same, 0.1 and 5. However, the medication reduces the duration of infectiousness to just 1 day. We want to know if this intervention will stop the outbreak. We use the exact same formula, but update the duration variable to reflect the medical treatment. We multiply 0.1 by 5 and then by our new duration of 1. The final answer is 0.5. Because this value is now less than 1, the infected child will not even infect 1 whole person on average before being cured. This proves mathematically that reducing the duration of infectiousness successfully breaks the chain of transmission. To solidify our understanding, let us look at this transmission cycle diagram. In the center, we have an infected human host, highlighted in red. Notice the arrows pointing outward toward the susceptible hosts in gray. These arrows represent the pathways of infection. Along these pathways, you can see our key variables, beta, C, and D. The diagram visually reinforces that the contact rate brings hosts together, the transmission probability dictates if the jump is successful, and the duration determines how long this window of opportunity stays open. At the top, we see the condition R not greater than 1, reminding us that when these factors combine powerfully enough, the central host will successfully infect multiple others, driving the epidemic forward. Now that we understand parasites, let us shift our focus to a much more lethal cousin, the parasitoid. Imagine a creature that lays its eggs inside a living host, and when the babies hatch, they consume the host from the inside out. While a standard parasite wants to keep its host alive, a parasitoid's ultimate goal is to consume and destroy its host completely. Typically, these are insects like specialized wasps or flies. Why should we care about such gruesome creatures? Because they are some of the most important allies humans have in agriculture. Since they are highly specific and always kill their hosts, they act as natural pest control agents. Without them, crop-destroying caterpillars would cause massive global food shortages. Farmers actually release them as a safe alternative to toxic pesticides. To use parasitoids effectively in agriculture, ecologists use a mathematical model to calculate exactly how many pests will be destroyed. The formula is NP equals NH times the quantity 1 minus E to the power of negative A times P. Let us break down these symbols. NP is the number of hosts that are successfully parasitized and will die. NH is the initial number of pest hosts in the field. The variable A represents the attack rate, or how efficiently the wasps search. P is the density of adult parasitoids released. Finally, E is the base of the natural logarithm. Notice the negative exponent? As the attack rate or the parasitoid density increases, the negative exponent becomes larger in magnitude, making the E term smaller. This means the fraction inside the parentheses approaches 1, resulting in a higher number of dead pests. Let us apply this model to a real farming scenario. A farmer has a greenhouse infested with an initial population, NH, of 1,000 caterpillars. They release wasps, creating a parasitoid density P of 10 wasps per square meter. The search efficiency A is 0.05. We need to calculate how many caterpillars will be killed. We use our parasitism equation to find the number of doomed pests. First, we substitute our values into the exponent, multiplying 0.05 by 10, which simplifies to negative 0.5. The value of E to the negative 0.5 is approximately 0.606. Subtracting this from 1 gives us the proportion of the population successfully attacked. Multiplying this proportion by 1,000 gives a final answer of 394. Physically, this means the wasps will kill nearly 400 pests, which is a good start. The farmer from our previous calculation wants better results, so they decide to scale up the biological control. They release more wasps, increasing the parasitoid density P to 40 wasps per square meter. The initial population and search efficiency remain the same. We want to find the new number of parasitized caterpillars. We apply the exact same mathematical model, updating only the density variable. In the exponent, we now multiply 0.05 by 40, which gives a much larger negative number, negative 2.0. The value of E to the negative 2 is approximately 0.135. Subtracting this from 1 gives a much higher proportion, of 0.865. Multiplying by 1,000 gives a final answer of 865 caterpillars. By quadrupling the wasps, the farmer mathematically ensures the destruction of the vast majority of the pests, saving the crop. Let us visualize this lethal biological interaction. On screen, you see a parasitoid wasp landing on a much larger green host caterpillar. Notice the specialized structure called the ovipositor being inserted into the caterpillar to lay an egg directly inside its body. This is the exact moment the attack rate variable from our formula is realized in nature. In the top right, there is a crucial inset graph showing an exponential decay curve. The x-axis represents the parasitoid density, and the y-axis represents the surviving hosts. As we proved in our calculations, as the density of wasps increases along the horizontal axis, the number of surviving caterpillars drops exponentially. This diagram perfectly bridges the gruesome biological reality with the elegant mathematical model that ecologists use. We have covered parasites and parasitoids. Now let us introduce the third crucial player, the vector. Think about a food delivery driver. They do not cook the pizza and they do not eat it, they just transport it. In infectious diseases, a vector is exactly that, a living vehicle that transports a disease-causing pathogen from one host to another. There are two main types. A mechanical vector, like a housefly, simply carries the pathogen on the outside of its body. A biological vector is much more complex. The pathogen actually lives and multiplies inside the vector's body, like malaria inside a mosquito. Because pathogens rely entirely on these vectors to travel, we often cannot stop the disease just by treating humans. The most effective public health strategy is vector control, eliminating the insects themselves to break the chain of transmission. To measure how intensely a vector-borne disease is being transmitted, scientists calculate the entomological inoculation rate, or EIR. Conceptually, this metric tells us exactly how much danger a local population is in by calculating the number of infectious bites a person receives. The formula is EIR equals M times A times S. Let us define these terms. The variable M represents the density of the vector, meaning the number of mosquitoes per person. The variable A is the human biting rate, which is how often a single mosquito bites a human per day. Finally, S is the sporozoite rate, which is the proportion of mosquitoes actually infected with the pathogen. If the mosquito density, the biting rate, or the infection rate increases, the EIR increases, meaning people are getting infected much more frequently. Let us calculate the danger level for a tropical village. Researchers find a mosquito density M of 10 mosquitoes per person. The biting rate A is 0.2 bites per day. Laboratory testing shows the infection proportion S is 0.05. We need to calculate the daily entomological inoculation rate. We use our EIR formula to establish a baseline of transmission intensity before attempting any interventions. We substitute the given values directly, multiplying 10 mosquitoes per person by the 0.2 bites per day and then by the 0.05 infection rate. The final answer is 0.1. Physically, this means a person in this village receives one infectious mosquito bite every 10 days. Over a year, this results in dozens of infectious bites, indicating a severe risk of contracting malaria. To protect this village, a health organization distributes insecticide-treated bed nets. The mosquito density M remains 10, and the infection rate S remains 0.05. However, the physical barrier of the nets drops the human biting rate, A, drastically to 0.01 bites per day. We need to see if these nets are effective. We use the same mathematical formula, updating only the biting rate variable to isolate the specific effect of the bed nets. We multiply 10 by our new, much lower biting rate of 0.01 and then by 0.05. The final answer is 0.005. This means a person would now only receive one infectious bite every 200 days. This massive mathematical reduction proves that simple interventions can drastically disrupt the vector's ability to transmit disease. Let us visually compare the two types of vectors we discussed. On the left side of the diagram, we see a mechanical vector, a housefly landing on food. Notice the bacteria on the outside of its feet. It is simply transferring the pathogen via physical contact, much like a dirty delivery vehicle. On the right side, we see a biological vector, a mosquito taking a blood meal. Look closely at the cross-section. The parasites are not just hitching a ride. They are actively multiplying and developing inside the mosquito's salivary glands before being injected into the human skin. This diagram clearly illustrates why biological vectors are so intimately tied to the life cycle of the diseases they carry, making them a primary target for global eradication efforts. We have reached the end of our journey. Let us review the key takeaways. First, parasites are masters of stealing resources without immediately killing their hosts, and we can predict their spread using the basic reproduction number. Second, parasitoids blur the line between parasite and predator by inevitably killing their hosts, making them nature's ultimate biological weapon for agricultural pest control. Finally, vectors are the living bridges that allow pathogens to cross from one host to another. Whether mechanical or biological, they are essential for the survival of many diseases. By understanding the mathematical and ecological dynamics of these organisms, we can design interventions like bed nets and deworming medications to save millions of lives. You now have a solid foundation in parasitology.